Click for IB Biology. Here we're looking at mutation, the production of single nucleotide polymorphisms, and the concept of genetic conservation. Much of this is a development of some of the items which were raised as we went through transcription and translation. There's a PDF, a blank PDF of this presentation down in the description. You can annotate that with your own notes. There's a 10-question quiz with answers and a channel link with an AI prompt which will allow you to set up your personalized study questions. Mutation. So we've previously met the idea of point mutations and the way in which such point mutations can, in one instance with substitution, we can see here that the G has been substituted with an A, and this changes a single amino acid. So the question is, what influence does this have on the protein and its function at the end of the day? The alternatives to substitution are deletion and insertion, and these tend to have more drastic effects. By deleting or inserting, what we do is we end up changing what is called the frame shift, the sequence of bases and the codes, the triplets and the codons that they make up. So let's just take a quick look at that as a reminder. We're going to delete G, and so now our codons will read CGC, UUU. So notice it's now stepping over into the third codon of the original messenger RNA, and this causes a total change in the reading of the amino acid. So we are predicting that this will have a drastic impact. And the same is true of insertion, where we put a base into the base sequence of the gene. It transcribes to this here, and frame shifting causes big changes, often introducing what are referred to as premature stop codons. It terminates the protein sequence, polypeptide sequence, very, very quickly. So the cause of these changes to the DNA are mutations, or that's what they are. These mutations are caused by mutagens, and mutagens take on a variety of forms, but we've summarized them to 4 here. The first one is chemical mutagens. An example of that would be the polycyclic hydrocarbons, which one finds in the tars of cigarette smoke. And these target the cells of the bronchial region of the lung, and often can end up mutating tumor suppressor genes like P53, and so the suppression of the tumor is released and the tumor develops lung cancer. Skin cancer is another one. With radiation from the sun, of course, UVB is particularly associated with inducing changes to the skin cells and tumor induction there. DNA replication, errors do occur in DNA replication, but when we were covering the study of DNA replication, we mentioned that there were corrective mechanisms, a multitude of corrective mechanisms for DNA which protect the cells, but even so, sometimes errors do creep through. Viruses are known to cause mutation. Viruses are actually the study of A2.3 in the syllabus for higher level students, where you will find how viruses are able to insert DNA into the DNA of its host, and these two can end up inducing mutations which can end up being developing into tumors. If you want to review tumors, then you should return to the section of the syllabus on the cell cycle and mitosis. So these single point mutations, what effect do they have? And so I've already pointed out when we did transcription, translation, that because of the degeneracy of the genetic code, mutations might mean that we change UUU becomes UUC, but we still end up translating phenylalanine. And the same could be said here for leucine. If originally we have UUA and it's mutated to UUG, then it still gives us leucine. And so there is no impact. And it gives us a kind of evolutionary buffer. So some sort of a buffer system here to the mutations which the environment causes in our genetic code in all organisms. But what about when they do have an effect? So we have a change in the base sequence due to the mutation, which then in turn changes the messenger RNA, which in turn changes the primary structure of the polypeptide. And of course, then when the polypeptide is shaping up, the shape of the protein will dictate its function. So necessarily we would argue that change in shape will result in a change in function. So change the gene, you change the messenger RNA, you change the primary structure, you change the shape, you change the function. Now, what is the consequence of changing the shape and function? Well, it is possible that a mutation could occur and we actually end up improving the protein. Let's say that we actually improve the mechanism of the active site of an enzyme. But one can imagine that there are many more ways in which one can reduce the efficiency of a protein, and indeed, for example, in enzymes, by mutating the active site, it may be that the enzyme is actually rendered inoperable. So positive and negative effects. And then there's always the possibility that there's a change. And whilst if we think of this in terms of the protein of an enzyme, let's say that rather than the active site, the mutation occurs out here in this body of the enzyme. Perhaps the mutation here has no effect on the operation of the enzyme. And so we have what is called a neutral mutation. Another way of looking at mutation is to realize that this is actually the source of the alleles of genes. So where students will be aware of the idea that a gene is often, there have multiple forms of that gene which are known as alleles. And whilst they code for the same characteristic, you have the gene for the same characteristic, the form of that characteristic can be changed by a change in allele. Those come about through point mutations, single mutations, and so we end up with single nucleotide polymorphism, which means that we have different base sequences for the same gene, and this is the source of genetic variation. So perhaps we have a point mutation that introduces antibiotic resistance into a bacteria, so we have then resistant forms and susceptible forms, as you'll have heard about with MRSA, methicillin-resistant Staphylococcus aureus, the flesh-eating bacteria associated with wound infection. The random errors in the genetic code, and they are random, there is no directional nature to mutations. They can occur anywhere. There's no place in particular makes an error happen somewhere. So they're random errors. And these are random errors in the genetic code create the variation upon which non-random selection can then select those organisms that either fare better, they have an advantage, or they have a disadvantage, which of course is the mechanism of natural selection. Let's just hesitate for a moment to just go through that again. Mutation is the source of the variation, of the genetic variation. This is random. But selection is non-random. Survival is not random. These two factors here are two of the major components of the principles of natural selection that we will study in a topic on its own. looking at DNA, what we find is that there are particular regions of DNA which are more susceptible than others to mutation. These regions tend to be between the nucleotides. So the nucleotide, the open chromatin between the nucleosomes tend to be more susceptible to mutation, whilst the genes curling around the histones are tightly held to the histones, restricting the effects of mutagens in those areas. And so vulnerability to mutation is often dictated by your position on the chromosome in relation to the packing nature of the DNA. When one looks across the variety of species and living things, it's remarkable how much conservation is of genetic sequences over evolutionary times. We're saying that genes have survived over times and are present in multiple species because they are so effective in what they do. Those genes are, in order to be conserved, tend to be ones associated with the nucleosomes as protection regions, and they tend to be associated with these correctional mechanisms. The details of these the student does not need to know. So highly conserved genes, ones which are found across the variety of living things, tend to have been protected and conserved. And the reason for this is because they are very, very often associated with the development of the organism from its early stages, even way back into morphogenesis and the development of the embryo, which we've already studied. The second video in this series is now appearing here. There's a chance to subscribe to the channel, and the playlist is listed on the right hand side for this particular topic.